Good evening. We are so happy that we are together today for the PSC Partners Roadmap Research 201 meeting. The PSC research landscape now and into the future, identifying research questions. My name is Mary Villas, and I'm the president of PSC Partners Seeking a Cure Canada. It is so exciting that so many have joined us this September evening. This meeting will start with a general session about 4 general areas of PSC research, followed by moderated breakout rooms for discussion on these areas of research. Next slide. Before we get started with this Roadmap 201 meeting, I'd like to briefly review what the Roadmap Initiative is all about. Roadmap is one outcome of work that has been ongoing over the past few years. This includes the receipt of the CZI Rare as One grant. A primary goal of this grant is to leverage the power of patients to accelerate PSC research, including the development of a patient-organized collaborative research network. The October 2020 PFDD meeting with PSC with the FDA, meeting on PSC with the FDA, and the PSC Partners Patient Registry, which currently has over 1,900 participants from around the world. Together, this work has led to, next slide, please, expanding the vision of the role of PSC partners and the PSC patient community in research and drug development. With that bit of context, let's revisit the goals of Roadmap, which are accelerating progress to find therapies and a cure by educating the patient community on the research landscape, broadening the researcher base, and strengthening multi-stakeholder communication, and creating opportunities for researchers and clinicians to engage in meaningful discussions with an informed patient community. Next slide, please. Practically, what does this mean? Hopefully, you've seen this roadmap graphic, which to the right shows the 3 main stakeholders. On the left shows the 4 topic areas as steps. We are currently on topic 2, research. For each of these topics, we are hosting a series of meetings with 2 or more Zoom meetings for each topic. The 101 sessions are community focused and educational. The 201 and 301 sessions are multi-stakeholder discussions. Next slide, please. Thank you so much again for joining us and welcome. Now I will pass the Zoom mic to Ricky Safer, founder and CEO of PSC Partners Seeking a Cure.
Hi, thank you, Mary. And thank you to all of tonight's attendees, both first-timers and repeat attendees. This is the second of 3 research webinars, as Mary explained. So our goal tonight is to present brief overview of those 4 important topics in current PSC research, and then in the breakout rooms to give PSC patients and caregivers an opportunity to engage in meaningful discussions with researchers and clinicians on these topics. Our agenda offers varied educational opportunities for today's multi-stakeholder attendees. During the general session, Dr. Lewis Roberts, Mayo Clinic, will speak about PSC genetics. Dr. Stephen O'Hara, also of the Mayo Clinic, will cover the microbiome. Dr. Paul Dawson, Emory School of Medicine, will discuss bile acids. And lastly, Dr. David Assis, Yale University School of Medicine, will discuss immune cell research. PSC Partners is pleased to have supported much of this research. I'd like to thank our roadmap sponsors, Alvareo Pharma, Intercept Pharmaceuticals, and Escient Pharmaceuticals for their support of our webinar series. Now, I'm pleased to introduce Dr. Lewis Roberts, who will speak about PSC genetics research. Thank you for joining us tonight, Dr. Roberts. Dr. Roberts is professor of medicine at the Mayo Clinic in Rochester, Minnesota. His research goals at the Hepatobiliary Cancer Laboratory are to understand the mechanisms by which liver and biliary cancers develop, grow, and spread, and to develop and validate methods for early detection, diagnosis, and treatment of primary liver, biliary, and pancreatic cancers. I'll turn it over to you, Dr. Roberts. Thank you.
Thanks very much, Ricky, and I'd like to thank PSC Partners for the opportunity to present this work, which is on genome-wide association study, which I will explain on bile duct cancer of which cholangiocarcinoma is a part. And as I'll explain, cholangiocarcinoma is one of the risk factors of PSC. And this is on behalf of a fairly large group of collaborators that have formed a consortium to try to better understand the genetic underpinnings of this disease. Next slide, please. And I have to recognize collaborators from a number of countries and different states in North America, several countries in Europe and Asia as well. Next slide, please. So biliary tract cancers include cholangiocarcinoma, which is bile duct cancer, and gallbladder cancer. And we typically will distinguish between intrahepatic cholangiocarcinomas, which form in the smallest of the bile ducts, so between what we call the second order divisions of the bile duct all the way up to the very edges of the liver. Then perihilar or hilar cholangiocarcinoma is the other cancers that develop in sort of the entryway into the liver. And so these are the major two branches, the right and the left bile ducts, as well as the common hepatic duct in the central part of the liver. And then we have what we call distal cholangiocarcinomas, which extend from where the gallbladder duct or cystic duct enters the bile duct into the small intestine. So the function of the biliary system is to collect the bile that's made by the liver. It gets stored in the gallbladder. When we eat a meal, particularly one that contains fat, it causes this valve at the bottom here to open up. The gallbladder contracts, and the bile, which helps to digest our food, is emptied into the small intestines. We call the two types of cholangiocarcinoma that form in this entrance to the liver as well as the distal area together extrahepatic cholangiocarcinomas. Next slide, please. So primary sclerosing cholangitis, or PSC, is a risk factor for biliary tract cancers. And it actually is a risk factor for all types of biliary tract cancers, but particularly cholangiocarcinoma, which is the bile duct cancers, and even more especially the perihilar cholangiocarcinomas. Patients with PSC over their lifetime have about a 15 to 20% risk of cholangiocarcinoma. And about a third of the cholangiocarcinomas that form in patients with PSC will be diagnosed in the first 2 to 3 years after diagnosis of PSC. And we suspect that this is in part because it's actually the early formation of a cancer that brings attention to the diagnosis of PSC. So after the first 2 or 3 years, PSC patients will develop cholangiocarcinoma at the rate of about 0.5 to 2% per year. Cholangiocarcinoma development appears to be determined by environmental, lifestyle, as well as potentially inherited factors. The fact that there are familiar tendencies towards risk factors such as gallstones, PSC, and PSC with inflammatory bowel disease suggests that inherited genetic predisposition may be a factor in the development of cholangiocarcinoma as well. Next slide, please. It's become quite clear that early detection of cholangiocarcinoma, as well, of course, many other cancers, improves patient outcomes. What I'm showing here is a staging system that we developed based on patients with cholangiocarcinoma that we had seen at Mayo Clinic from the very earliest stage 1 all the way to the most advanced stage 4. And what you can see here is that stage 1 cancers had the best outcomes compared to the other stages. So we know that early detection is important in improving patient outcomes. Next slide, please. And consequently, we have a number of different approaches to try to, interestingly, this test that we developed was able to actually quite effectively diagnose cancers that were de novo, that is, that formed in patients without PSC, but it was not as good as detecting cancer in individuals with PSC. So there's clearly differences in the biology between cancers that form on their own versus cancers that form in patients with PSC. Next slide, please. So the basis really for this study is the understanding that genetic risk factors for cholangiocarcinoma might help us to determine those who are at highest risk of developing cholangiocarcinoma and help us to improve early diagnosis. And this is in part, as I was saying, because the risk factors for cholangiocarcinoma involve many mechanisms which have underlying genetic predispositions. And we recognize that they may be influenced also by genes in different cellular pathways. And genome-wide association studies uncover common gene variants across the entire genome. So they allow us to study the entire genome of individuals who have cancer, compare them to the entire genomes of individuals without cancer, and try to identify whether there are specific variants in genes that are associated with a higher risk of cancer. Beyond the typical genome-wide associations that use what we call single nucleotide polymorphisms, or individual changes that are fairly common in the population, we can also do whole genome sequencing and family studies to allow us to identify rarer genetic variants that are associated with risk of cholangiocarcinoma as well. Next slide. So this shows an example over on the left side. We have individuals with disease, in this case, individuals with cholangiocarcinoma, and on the right side, we would have controls, individuals without the disease. And if you'll just point your attention to SNP 3 at the bottom, you'll see that here we have a single nucleotide polymorphism that where the red dots, which reflect a particular gene variant, are much more common in the individuals with disease than in the individuals without disease. So that SNP might be, in a sense, a marker for individuals that have an inherited predisposition to this particular disease, in this case, cholangiocarcinoma. Next slide, please. And so this, in part because cholangiocarcinoma is a relatively uncommon disease, this has been an effort that's really been a joint effort between collaborators from multiple institutions. And so this chart just gives you an idea. Many institutions in the United States, but also in Europe and Denmark, Sweden, Germany, and Canada as well, have collaborated together for this initial phase of this study. And we assembled a total of about 2,600 samples from patients with cholangiocarcinoma, and then about 660 samples from patients with gallbladder cancer for this initial study. Next slide, please. And this is the breakdown by country. And so the predominance of the samples came from the United States, but you can see we had contributions from a number of other countries as well. Next slide, please. So this is the initial analysis that we did for this group of patients, and we broke it down into those with intrahepatic cholangiocarcinoma versus those with extrahepatic cholangiocarcinoma. These are the patients of European descent that had intrahepatic cholangiocarcinoma. And what you can see here in the graph on the right is you see the two lines that cross. There's a blue line, and then above it, the red line. And what these lines are represent the, in a sense, likelihood that there's a genetic predisposition by one of these dots, which is one SNP in one of the chromosomes, one of these single nucleotide changes in a gene. Now you can see that some of these dots, the ones that have arrows pointing at them, rise above the blue line, but they don't quite reach the red line. So that tells us that they are suspicious as being regions that might be associated with cancer, but they don't quite meet the threshold where we say they are definitely associated with cancer. So clearly for this part of the study, we need more samples because in general, more samples gives you a higher likelihood of being able to find these abnormal regions. Next slide, please. And this is looking at the extrahepatic samples. And so this includes the patients with the perihilar cholangiocarcinoma that is more typical of PSC associated cholangiocarcinoma. And you can see here that, again, most of the dots we see are above the blue line, but don't quite reach the red line. There's a single region, though, the sort of purple arrow, where you can see that the dots rise up above the red line. So that's a highly suspicious region. But again, only one of these regions reached significance. And so the hint there is we do need more samples to get a better confidence in the results. Next slide, please. And so what we are doing is expanding the number of centers. So we are now up to, I think, around 30 centers that have agreed to contribute to this next expansion phase of the study. We are very interested in having individuals and patients collaborate as part of this study. And I put my email address there if you have any interest at all in participating. We are also collaborating with the Cholangiocarcinoma Foundation. So if you go to the Cholangiocarcinoma Foundation website, there's actually a link for patients who are interested in participating in the study, where they can send a note to us and then we'll contact them for participation. Next slide, please. So I'd really like to acknowledge the support from PSC partners, from the Cholangiocarcinoma Foundation, and from the network, that's the International Hepatobiliary Neoplasia Repository. Next slide, please. And again, there's my email address if anyone has questions. Thanks again for the opportunity to present.
Thank you so much for sharing your fantastic work with us, Dr. Roberts. We'll get to Q&A in the breakout rooms. Our next topic is the microbiome. And Dr. O'Hara, thank you so much for joining us. We look forward to hearing your presentation.
Thank you, and thanks, Louis. That was really important work and very informative. And thanks to the organizers for inviting me to give a brief overview of the microbiota and PSC. The influence of the gut and the microbes that reside therein in PSC has been an area of interest for decades, stemming from the realization that a high percentage of PSC patients also have inflammatory bowel disease. Next slide, please. The gut-liver axis refers to the bidirectional communication between the gut and the liver. Blood flow from the gut via portal circulation brings dietary constituents, environmental chemicals, bile acids, microbial components, etc., to the liver and influences liver function. Conversely, the liver alters the gut environment through the release of bile, which contains bile acids, cholesterol, phospholipids, etc. And hence the liver and the gut are in constant communication. Next slide, please. Alterations to the normal composition of the human gut microbes, or dysbiosis, can have important implications for inflammatory processes, metabolite production, and communication with essentially all other systems in the body. Given the likely influence of the gut on PSC, the composition of the gut microbiota was assessed. And the terms microbiome and microbiota are technically different, but are most often used interchangeably. And I apologize in advance, I'll probably use them interchangeably throughout this talk. Microbiota specifically refers to the assemblage of microorganisms present within a defined environment. For example, bacteria in stool. Many studies asked what bacteria are present in PSC patient stool and if this differs from healthy controls in patients with IBD. Frequently, the technique that is used to answer this question is PCR amplification of bacterial 16S ribosomal RNA gene, specifically the variable region of this gene, or the region that differs between bacterial types. This is amplified, sequenced, and aligned to known bacterial sequences or reference genomes to identify the bacteria present in the sample and determine their relative abundance. This information is used to evaluate the diversity of that population. That is, are there a lot of different types of bacteria present, a concept known as species richness, And does one or several of the bacterial types dominate over the others, or a concept known as evenness of the population? What was found in the PSC patient population is that alpha diversity, the diversity within the PSC patient samples, was reduced. And between sample diversity, or beta diversity, that is between the PSC patient and healthy control populations, that was different between the two. One interesting finding that is becoming a little more clear from these studies is that the liver disease appears to be a dominant driver of changes to the bacterial composition. That is, the presence of colitis only had a minor effects in PSC patient microbiota composition. Several smaller studies have also indicated that the bile duct microbiota of PSC patients differs from controls as well. To expand on this further, the analysis of 16S ribosomal RNA demonstrated that the microbiota was different, but metagenomic shotgun sequencing addresses not only who is there, but what they might be doing, functional metagenomics. This technique amplifies all the genomes and genes that are present in a particular environment. So not just the 16S bacterial DNA, but genes and enzymes, transporters, et cetera, that can be further analyzed for understanding the metabolic capability of a community. A recent study that just came out demonstrated that PSC patient fecal samples exhibited fewer microbial genes overall compared to the healthy controls. And when the genes present were further analyzed using metabolic pathway analysis, it was discovered the differences existed in the abundance of genes required for vitamin and amino acid metabolism. To take this to the next level, you can identify what molecules are present using metabolomics. So while metagenomics would predict that a certain subclass of molecules may be present or absent based on the genes present, metabolomics would determine whether these are actually decreased or increased in a biological sample, for example, in serum. To do this, one would use mass spectroscopy approaches. In the same study that I mentioned about the metagenomics, it was found that not only did the metagenomics predict differences in amino acids and vitamin composition, but found reduced amounts of these in patient serum. As with all good science, of course, this observation needs to be confirmed through further studies, but I believe it's a step forward in not only understanding what microbes are present or absent, but what this may mean physiologically. In work previously supported by PSC partners, our group looked at the biometabolome in PSC patients and healthy controls. We also found difference in the bile composition, and I'm very excited to go back and re-examine our data set to compare what Kuman et al. Had found to see if I can formulate any new hypotheses of what's maybe going on. Next slide, please. Importantly, and I think justifiably, many hypotheses testing disease progression in PSC are framed around this gut-liver axis. I'll just touch on each of these just very briefly. First, bile acid homeostasis. Intestinal epithelia respond to bile acid composition in the gut, a process that is influenced by microbes. The transporters and receptors found in the gut epithelia not only recycle these bile acids back to the liver, but also initiate signaling through this FXR, this bile acid receptor in the intestinal epithelia, secreting a peptide hormone back to the liver to suppress further bile acid synthesis. The leaky gut in the bottom left, a primary function of intestinal epithelia is to keep microbes and potentially injurious molecules contained in the intestine. That is, it performs a barrier function. A variety of factors can influence a breakdown of this barrier function, including certain microbes, as was recently demonstrated, a couple of years ago demonstrated. Once this barrier is breached, there can be, of course, a localized immune response, but additionally, some of these microbes or microbial products or other contents from the gut may be brought back to the liver and influence liver function. In the upper left, we have aberrant homing. That is, immune cells that are primed in the gut should stay there. But in disease states, the molecules on blood vessels that direct these cells to the gut are aberrantly expressed in other tissues, including the liver. And hence inflammatory cells that should remain localized to the gut may accumulate in the liver and result in inflammation. And finally, there's the bad bile or leaky bile hypothesis, and this posits that the composition of bile, which is influenced by what is coming from the gut, may influence the cells that line the bile ducts, or the cholangiocytes as they're called. And as with all epithelia, a primary responsibility of this epithelia is to function as a barrier. And if this is breached, inflammation may ensue. Our group also spends a lot of time trying to understand how the bile duct epithelial cells sense and respond to the composition of bile. We're particularly interested in a cellular state known as senescence, where a cell permanently stops dividing and it starts secreting pro-inflammatory signals. Another concept that is studied quite a bit is the bicarbonate umbrella. Cholangiocytes secrete sodium bicarbonate, which influences bile flow, but it also forms a buffer between the bile content and the membranes of the bile duct epithelia. Next slide, please. So just briefly, I'll give some examples of therapies that either directly or indirectly target the gut microbiome. First is bile acid regulation. The idea, if you can activate this bile acid receptor, FXR, you could slow the secretion of bile acids from the liver through this gut to liver axis. The next that is directly targeting the microbes in the PSC patient is fecal microbiome transplant. And as you might guess, this is taking stool from a healthy patient in the attempt to repopulate a dysbiotic microbiome for therapeutic benefit. And thus far, there's been a single case report as well as a small pilot trial that have been published. And importantly, this pilot trial did demonstrate that it was safe, and it also showed that it did have improvements in one of the endpoints, alkaline phosphatase, in at least a subset of the patients. Our group is interested in the oral delivery of select freeze-dried microbes as a potential therapeutic, and I currently have a grant that's under review with Alexander Korutz from the University of Minnesota. And Alexander has been really a pioneer in pushing forward this concept of not only fecal microbiome transplant, but potentially a select number of microbes. In particular, he's done this in Clostridium difficile infections. There's also been a lot of interest in antibiotics. A lot of this interest has stemmed from smaller studies and maybe perhaps some anecdotal evidence. But vancomycin is an antibiotic, a glycopeptide antibiotic that targets gram-positive bacteria. It's not certain whether this functions directly by altering the gut microbiota or whether there's a component of immunomodulatory effect or a combination of the two. And I just wanted to point out that there's a study going on right now with my colleagues here at Mayo, and this is still an active trial and they are recruiting. And if you'd like more information on that, you can feel free to contact Dr. John Eaton at that email there listed there. There's also liver inflammation, nor-ursodeoxycholic acid or nor-UDCA. This is a modified ursodeoxycholic acid molecule. And it's been reported that this not only promotes bile flow, but it also promotes the maintenance of this bicarbonate umbrella, this protective layer that protects the epithelial cells from bile. And we are also interested in what's known as senolytic therapies. This is small molecule targeting of senescent cells for therapeutic benefit. We've completed preclinical trials in animal studies, and we're looking to move this forward in a small pilot study within the next year or two. Next slide, please. So in summary, the gut and liver are in constant communication. The gut microbiota in PSC patients differs from healthy controls. The molecular output from PSC patient microbiota likely differs from healthy controls. Gut microbes influence several potentially pathologic processes, and the therapeutic potential of microbiota manipulation in PSC is being addressed clinically. And so in conclusion, our understanding of microbes and the gut-liver axis has vastly improved just over the past several years, and several promising therapeutics related to this axis are currently being evaluated. And that's what I have for you tonight. Thank you for allowing me to speak to you tonight.
Thanks so much, Dr. O'Hara, for sharing this really comprehensive view of the gut-liver axis. Our next topic is bile. Dr. Dawson, thanks for joining us tonight, and please take it away.
Okay, so my task this evening is to give a high-level overview of bile acids and then connect the function of these interesting molecules to the to PSC. Next slide, please. I'm going to provide a brief introduction to bile acids, to talk some about their function so that you understand. Several of the speakers have already mentioned bile acids and their connection to PSC. I'll talk about bile acids in general and their connections to liver disease, and then mention some bile acid-based therapeutics. And shown on this first slide here is just a general structure of a bile acid, which is, if you can, next slide, please. One back, please. So bile acids are, you can look at these, you can see that they're, you can recognize that these are small steroid-like molecules. I tend to think of them as the superheroes of small molecule endobiotics, the endogenously synthesized compounds. And what you can appreciate from this slide here is that there's a basic 4-ring structure, and then there's diversity in terms of the overall structures of these of the different bile acids. So when we talk about bile acids, we're not talking about just one molecule. We're actually talking about quite a large family of molecules which can impact their function. Next slide. So what are bile acids? So bile acids are the major component of bile. So on the left shows the liver. Bile acids are synthesized from cholesterol and hepatocytes and secreted into bile. And the liver makes and secretes bile continuously, but the flow into the duodenum is intermittent because some of the bile is diverted into the gallbladder during the interdigestive period. And the liver is amazing in terms of its ability to produce bile. We could produce as much as a liter of bile or more per day. And if we look in bile, what's present there is that it's mostly water, but the major solute that's present in bile are these bile acids that I mentioned earlier. There's also a number of other lipids, fats that are present. There's phospholipid and cholesterol and a variety of other constituents. And bile acids that I'm discussing now is an end product of cholesterol. Cholesterol is a very oily, hydrophobic molecule, and then through a complex set of reactions is converted to a water-soluble molecule here that shares some similarities in terms of its basic steroid structure. Next slide. So one of the major functions of bile acids is to drive bile flow. And in work from Jim Boyer, someone here on the panel this evening, and others, the secretion of bile and bile acids into bile is one of the major driving forces for the movement for the production of bile. The other major function of bile acids is that they play a critical role in the digestion and absorption of fat, particularly cholesterol, fat-soluble vitamins, and long-chain fatty acids. And that's because of their amphipathic nature. And then here's this basic space-filling diagram here of a bile acid. There's a very nice hydrophobic side here, this hydrophilic side here. And even though it's water soluble, this amphipathic nature makes bile acids good detergents. And as I showed, there was a line in an earlier slide, with great power comes great responsibility. Because the bile acids are detergents and play a critical role in solubilizing fats present here in the gut, when bile acids are trapped in the wrong compartment, they have the potential to interact with membranes, begin to solubilize membranes, intracellular organelles, and potentially contribute to a cellular and organ injury. So next slide, please. So what are the functions of bile acids? The major functions, as I said, were driving bile flow, its secretion by the hepatocyte into bile, solubilizing fats and fat-soluble vitamins. But over the past 20 to 30 years, we've identified a whole myriad of functions associated with these really interesting molecules. These these are sort of relatively conserved structures here. We know they're important for eliminating cholesterol from the body, that they can regulate gene expression by interacting with specific receptors, these so-called nuclear receptors, such as the farnesoid X receptor, the pregnetin X receptor, or vitamin D receptor. They also interact with another cell surface receptor called TGR5 and can regulate a variety of physiologic processes and they have whole body functions in terms of regulating metabolism. Within the liver and hepatocyte, they play important roles, as I said, in maintenance of bile flow. Liver endothelial function, there's interactions, and then in particular, they have important roles in the biliary tract lumen in keeping everything solubilized. Next slide, please. So to continue on this theme of bile acids, these really critical small molecules, we know that they can function to stimulate bicarbonate secretion, which is important as participates in the bicarbonate umbrella that we just heard about. They have important roles in regulating, they control the solubilization and absorption of dietary lipids, and especially, as I said, are really critical for fat-soluble vitamins and cholesterol. But they have this also complex interaction with the microbiome. That the bile acids will in part shape the structure of the microbiome, and in parallel, the bacteria, the microbiome in our gut, have the ability to take up and metabolize bile acids, converting them into a whole variety of different structures. And just like as in the hepatocyte, they can regulate expression of genes in the gut through these nuclear receptors or through TGR5, this other really important receptor that's present in a variety of different cell types and tissues. So bile acids have they're detergents, but they're also really important signaling molecules. And one of the other, and then in the next slide shows one of the other really important properties of bile acids. Next slide, please. And that bile acids themselves are and this one I titled "Not All Bile Acids Who Wander Are Lost." Bile acids are synthesized by the hepatocyte in the liver, secreted into bile, and then after enter the gallbladder, and then empty into the small intestine, where they have this important role in digestion. But then bile acids, the fat is absorbed in the proximal part of the intestine. Bile acids are absorbed in the distal part of the intestine and then carried back to the liver to be resecreted and reutilized. So the bile acid pool in humans is roughly 2 to 4 grams in size, but will cycle multiple times in response to a meal. And so the liver may see as much as 20 grams of bile acid sort of fluxing through it at any one time, which is a considerable amount of a detergent. So it's potentially, so this system, this complex enterohepatic circulation that's evolved it's one of its major functions is to provide bile acids to solubilize lipids when needed, but it's also to compartmentalize and to keep these bile acids in the correct compartments. And you can see that here, that in plasma, the bile acid concentrations are quite low, less than 5 micromolar. But in gallbladder bile, for instance, the bile acid concentrations are thousands of, 1,000 times or more higher at millimolar concentrations. So within the body, we have to maintain this balance between high concentrations in some compartments and low concentrations in the other. Next slide, please. And to make this more complicated, the microbiome has the ability to metabolize and alter bile acids. And so as a result, we get this large group of different structures, just like this large group of superheroes here, in response to the metabolism by both the gut microbiota as well as the host. And even though these molecules will function to different degrees in terms of their ability to solubilize fats and also signal. Next slide, please. So do they have a role in liver disease? As you might expect, a high concentration of a detergent is not a good thing to be in the wrong compartment. And there's abundant evidence from other forms of liver disease that the in part because of the detergent nature and also the signaling abilities of bile acids, that in conjunction with other hits such as infection, environmental, as well as genetic factors, bile acids will play a role in damaging the epithelium in the biliary tract and leading to recruitment of immune cells and perpetuating this hepatocyte and cholangiocyte damage. But not only do they play a role in liver disease, but in the next slide, and this is my last coming up, that bile acids may also have be part of the solution. So that there are a variety of different bile acid-based therapeutics in use or in development that can function to either inhibit hepatic bile acid synthesis, stimulate bile secretion from the liver, detoxify hepatic bile acids, or block intestinal or hepatic bile acid uptake. Some of these drugs, such as ursodeoxycholic acid, has been around for many, many years. Others have recently been approved for other forms of liver disease, such as FXR agonist and ileal bile acid transporter inhibitors. And so there's intense interest in whether these same approaches will have use for PSC. And we're very excited that whereas bile acids may be part of the problem, they may also be part of the solution. And last slide is, so thank you so much for your attention. And I'd love to talk more about my favorite molecule, bile acids. Thank you.
Thank you so much, Dr. Dawson. We love your enthusiasm and your really informative presentation. Our final presentation topic tonight is immune cells. Dr. Assis, thank you for joining us.
Hello, and thanks everyone for your attention, and thank you for PSC Partners for putting this great program together. It's a real honor to be able to contribute to this session today when I'll be speaking about the role of immune cells in what's wrong in PSC. Next slide, please. So I would submit that part of the glue, unfortunately, the glue of what causes a lot of these different facets of PSC to come together in a pathologic way really is the immune system. You can see in this very comprehensive slide from the Carlson group in Oslo that there are multiple things going on in PSC that we don't like. We have some genetic factors and abnormalities. We have some as yet fully unknown environmental triggers. We have a lot of what's going on in the gut, in the colon, in particular in colitis that can affect the bile ducts. We have toxic bile acids, as Dr. Dawson just mentioned. We have the development of scar tissue, fibrosis, and ultimately the risk of cirrhosis, liver failure, but also cholangiocarcinoma. And if you think about it, each of these facets, there is a key contribution of the immune system and immune cells in organizing, orchestrating, and perhaps even directly damaging the bile ducts and leading to a lot of these problems that we know and are trying to solve. Next slide, please. In fact, if you take a look at a GWAS or genome-wide association study from various autoimmune conditions, and I have in the top left corner there PSC, in terms of the mutations, what's wrong in genetic studies with PSC looks remarkably similar to the graphs, as you can see here, for what's wrong in rheumatoid arthritis, psoriasis, celiac disease, multiple sclerosis, and type 1 diabetes, suggesting very strongly that for all of the very unique aspects of PSC in terms of colitis, bile acid abnormalities, it seems to fundamentally share an autoimmune property in which the immune system seems to be genetically predisposed to a degree to cause some of this injury. Next slide, please. We know that the cholangiocyte or the bile duct cell is always talking. There's an ongoing conversation with the immune system. And in this cartoon, you can see that whether it's T cells, natural killer cells, mucosal invariant cells, and multiple cells from the immune system, they're always talking to the cholangiocyte. That happens in a more benign way in health, but certainly in the context of disease, the immune system is responding to distress signals from the bile duct cell, the cholangiocyte. Sometimes it is directly initiating a conversation and causing problems, but ultimately this crosstalk between the immune cells and the cholangiocyte is a big factor in some of the signaling that ultimately leads to the scar tissue and even the cancer risk that we know to be true in PSC. Next slide, please. And very importantly for this discussion, I would submit that the adaptive immune system, in other words, the part of the immune system that can learn and organize and react uniquely to environmental and other triggers, and also in an autoimmune way, is what's wrong in PSC. I showed that the GWAS studies a minute ago that there is a risk of genetic abnormalities that are common to other autoimmune diseases, such as the HLA loci. And very importantly, and something that we know and have to deal with, PSC can recur after transplantation. If you think about it, why is that happening? Why is a set of bile ducts, a new liver, being attacked by the body after transplant? Well, to a large degree, along with other factors, the immune system is perhaps recognizing once again another bile duct system and causing the disease. And lastly, I would submit that the predominant cell type, among others, in the portal inflammatory infiltrate and liver biopsies of these patients is the T cell, which we'll talk about a little bit further. Next slide, please. In fact, this recent article just published in Science Translational Medicine was a very comprehensive study of what's wrong in the bile fluid in terms of the immune cells in the tissue and in the blood of patients with PSC. And you can see here in this very busy slide, though, but some key subsets of T cells called CD4 T cells and CD8 T cells have an abnormal, and I would submit, a bit more toxic and aggressive profile in patients with PSC compared to patients who do not have PSC. And in the bottom half of the graph, you can see that certain key molecules that are secreted and also can instigate the immune system are quite highly present in PSC, in particular interferon gamma, IL-2, but very importantly, IL-17 and TNF. And we're going to talk about two of these in particular when we get to the second part of this brief presentation. Next slide, please. I won't spend any time on the liver-gut axis, which was eloquently described by Dr. O'Hara, just to point out that when the gut is triggered and there is an abnormality, as he had also mentioned, it's the immune cells, unfortunately, that get primed in a pathologic way in the lymph nodes as well as in the gut, and then travel to the liver and can be a part of the problem in initiating and perpetuating some of the damage and inflammation that occurs in and around the bile ducts in PSC. Next slide, please. I'd like to zoom in on interferon gamma for a minute. This is a molecule that can cause immune-related damage. It can instigate T cells to cause more damage in the context of PSC. Recently, there's been some articles not only in mouse models, but also in human studies showing that there is an increase production and role for interferon gamma in instigating the pathophysiology of PSC, as well as some molecules that are known to be related to the signaling pathway, including a recruitment molecule called CXCL10. And if you think about it, despite the fact that we're learning more and more about the role of the immune system in PSC, as of yet, there is no adequate way to modulate the immune system. In other words, how to harness the immune system to treat PSC. I would submit this is one of the big unmet needs. And this, as well as some other key pathways, deserve a lot more attention in how we can retrain the immune system to halt some of this damage and hopefully promote some healing. Next slide, please. Another facet of the T cell and the immune system in PSC, which is very interesting to me, is this harmful form of T cell called Th17 cell, which is a subtype of CD4 T cells. And this very interesting article and series of articles, I would say, from the group of Dr. Schramm in Germany, you can see here that these Th17 cells can react more aggressively, I would say, to exposure and stimulation with certain bacterial and even fungal products that are known to be present in the bile ducts. In other words, there seems to be some way in which the T cells in PSC patients negatively interact with some of the microbiome products that they're exposed to. So it seems to be a synergy between what's in the environment and what the T cell brings uniquely to the context of PSC. Next slide, please. And in fact, not only are there an increased negative risk for reactions between these Th17 cells and pathogens, bacteria and fungi from the microbiome, including the bile fluid, but there seems to be just more of these pathogenic T cells also in patients with PSC. And very interestingly, the same group, again, from Dr. Schramm in Germany, has shown that whether or not the patient with PSC has colitis, there seems to be an increase in the number and the pathogenicity of Th17 molecules and in this very harmful product called IL-17. Next slide, please. Last but not least, though, if we take a more broad view beyond just the T cell, I mean, some beautiful work by our co-moderator, Dr. Heather Francis from her group has shown that the mast cell also seems to contribute to some of the damage and even the production of factors that can make the T cells more pathogenic. And in particular, the mast cell, which can release histamine, has been shown beautifully in her group to play a role in PSC, as well as other liver disorders, and may be part of what we need to address in order to make the inflammatory environment and the T cell itself less reactive. Next slide.
Excuse me, Dr. Assis, this is your 2-minute warning. Thank you.
Sure. So we have an evolving pathophysiologic understanding of PSC in which, to a large degree, the immune system is responsible for a lot of the damage that we see in this disease. And frankly, I think we need to pay more attention to how to harness the immune system and some of these immune cells in the context of what we know to be going on in the liver. Next slide, please. Next slide, please. So one approach to looking at this, not only for the bile ducts, but also for the immune system, is through understanding and using organoids. And organoids are three-dimensional structures called organs on a dish. And these are an innovative way of studying a variety of diseases, including cystic fibrosis and cancer. These are self-organizing cells that can be developed from human organs and stem cells. And we are using this through a project recently funded by PSC partners to understand how the immune system interacts with the bile duct. Next slide, please. We have found that bile, which can be collected at the time of ERCP, is a rich source of cells that can be used to make organoids, these three-dimensional structures. And we've also found that these organoids can be used in order to lead to a reaction, an inflammation that can be studied through interaction with T cells. Next slide, please. We found that these organoids react to T cell generated products such as IL-17, as well as TNF alpha and interferon gamma. And we believe that these organoids from PSC patients can interact with PSC patient T cells in order to help us understand the dynamics of what's wrong with the immune cell and the bile duct. Next slide, please. So through this study, which I will briefly summarize, we have some insight and I believe a pathway to better understand how the immune cell, which is abnormal in PSC, interacts with the bile ducts. And I think thus far we have a lot to learn in how to use the immune system and the techniques that are available to us for this study without needing to rely on explant late stage tissue. Next slide, please. So to summarize this study, you can see that the T cells are very closely interacting with and clustering around the bile-derived organoid, or BDO. I mean, we see this in our in vitro models, and we also see a significant production of immune-secreted cells from the T cell, as well as the organoid recruiting inflammatory molecules. And very importantly, in some very early studies, it appears that the patient's T cell, when co-cultured with the patient's bile-derived organoid, lead to a significant increase in the amount of inflammatory molecules, suggesting that when you can study the bile duct cell and the T cell from the patient, there may be a possibility of understanding why this connection goes wrong in the context of PSC. Next slide, please. So therefore, this, among other beautiful work that's being done around the world, is seeking to understand how PSC and the immune system are integrated. This one approach which we're taking is to look at this co-culture of T cells and immune cells, other immune cells and bile-derived organoids from the same patient. Next slide, please. We have not yet made significant progress, sufficient progress in understanding therapeutically how to use this, but through this example, there is a possibility of reducing the inflammation through treatment with molecules that can inhibit the immune system's reactivity. Next slide, please. For example, a medication which is currently or recently been studied through a clinical trial called vidofludimus, which reduces and blunts the T cells' inflammatory properties, was studied in the context of this co-culture, and it significantly reduced the ability of the immune system to carry out the damage that it was predisposed to do on that bile-derived organoid. Next slide. So in conclusion, I would submit that it's important for us to think further about the immune system in the context of everything else that is wrong in PSC, because it's fundamentally an autoimmune disease with an abnormal immune function that recurs after transplant. Organoids and other similar models might be a way for us to better understand what exactly is wrong in the interaction between the immune system and the bile duct, and hopefully can lead to better treatment options, even through a personalized approach. Thank you.
Thanks so much, Dr. Assis. I'm sure we'll have lots of questions about organoids and your research in the immune cells breakout room.
I would love to thank tonight's speakers and our research facilitators for making tonight's webinar a success. I know our breakout room was wonderful. We had great questions and answers. So thank you also to all the PSC patients and caregivers, medical professionals, and industry representatives who participate in the breakout room discussions. In advance of our June 2022 conference, where we will launch our International Collaborative Research Network, these webinars are an educational venue to encourage open discussions between patients, caregivers, and researchers. If you missed our previous research webinars, feel free to go to the roadmap page on our website to listen to 2 recordings. One is our Research 101 webinar presentation last month, where Dr. Heather Francis covered 5 PSC research concepts. And the other was last week's casual Zoom chat session, where Dr. Ruth Ann Pye encouraged patients and caregivers on how to formulate research questions. Here are reminders about our current and future programs of interest. Please visit our website at pscpartners.org to learn more. Please save the dates June 2nd through 5th, 2022, and plan to attend our conference in Boston. We're planning this as an in-person gathering if the pandemic does not ruin it again. We'll be combining our traditional PSC patient caregiver conference with the addition of an International Collaborative Research Network component. And after 2 years of virtual conferences due to the pandemic, you won't want to miss this one. Join the patient registry if you haven't already. Ideally, we would love to have every PSC patient participate to help us accelerate PSC research. Visit our links to our updated COVID-19 and vaccine information. Sign up for a mailing list and follow us on social media. And now I'd like to turn it over to Ruth Ann Pye, who will give you a brief overview of next month's Research 301 webinar. Ruth Ann?
Thanks, Ricky. So one last quick update. We're actively prepping now for the Research 301 on October 26th, and we want to emphasize that your feedback through our post-webinar survey is highly valuable and appreciated. Please send in your thoughts and ideas for what you'd like to see and discuss during the 301 through this survey. And also feel free to send in questions that you didn't have a chance to ask tonight. A link to the survey will also be emailed to you shortly, but you can also follow the link here or scan the QR code on this slide. We're so excited that you all joined us tonight and to be continuing this important work to build towards our understanding of the research landscape in PSC. Please join us next month as we discuss the relationship between these PSC research concepts and continue asking research questions. Please keep an eye out in our monthly newsletter, social media pages, and website for communications with more details. We hope to see you all at the Roadmap Research 301 on October 26th. Have a great night, everybody.